All right, let's dive into something pretty incredible today. We're talking about a tool that's helping scientists unlock secrets that have been hidden in plain sight, right inside our very own genes. I want you to imagine something for a second. What if the cure for a disease, say cancer or Alzheimer's, wasn't some brand new drug, but a secret that's already in front of us, just buried in an absolutely impossible mountain of genetic data?
Well, you know what? That isn't some hypothetical question. It is the exact situation researchers are facing right now. This is what we call the genomic data challenge.
So, thanks to some mind-blowing technology, we are just flooded with genetic data. The specific term is transcriptomic data, which is basically a snapshot of which genes are switched on or off in our cells at any given moment. But here's the crazy part. This flood of information has actually created a massive bottleneck. We can generate the data way, way faster than we can analyze it.
And this bottleneck exists because the traditional tools for analysis are honestly a huge pain. First off, you pretty much need to be an expert programmer to even use them. On top of that, they're often super rigid. Maybe they only work with one specific kind of data. And of course, they demand these powerful, really expensive computers. All this basically locks out the very people who need the insights the most, the biologists and clinicians on the front lines of research.
So, with this massive challenge staring them in the face, researchers obviously needed a much better way forward. And that brings us to the hero of our story today, a brand new tool called DGEAR. So, what exactly is DGEAR? The full name is Differential Gene Expression Analysis with R, but forget the jargon. In simple terms, it's a super user-friendly, web-based tool that has one clear mission: to make powerful genetic analysis accessible to everyone, not just the coding geniuses. The way DGR works is by taking this ridiculously complex process and making it simple. It walks a researcher through the whole thing, from uploading their raw data on the left to doing all the heavy lifting analysis on the server, and then it delivers these crystal clear visualizations on the right. The whole workflow is just designed to be intuitive from beginning to end. But how does it do that analysis so well? I mean, what's the secret sauce here? Well, it all comes down to something pretty cool: the power of voting. Now, under the hood, DGEAR is built on this really solid foundation. You can kind of think of it as having three layers. There's the friendly user interface, that's what you see in your web browser. Then there's the powerful application brain on a server that does all the hard work. And finally, there's a secure database memory that keeps everything safe. This structure is what makes the tool so reliable and robust. Okay, so we've got thousands upon thousands of genes. How in the world does DGR sift through all of that to find the few that are actually important for a disease? Well, it uses a really clever strategy. It asks a panel of expert judges. This is called the ensemble voting method. Here's how it works, and it's brilliant. First, you have multiple different statistical tests. These are our judges, and they all analyze the data from their own unique angles. Then, each test, each judge, gets to vote on the genes it thinks are important. And here's the key: a gene only gets confirmed if it gets a majority of the votes. That ensures the final result is incredibly reliable.
So why does this matter so much? Well, any single test, any one judge might accidentally flag a gene that's just statistical noise, a false positive. But this voting method forces them to find consensus. It focuses only on the intersection you see here, the genes that multiple tests all agree on. That's how it filters out all the noise and really hones in on the candidates that are most likely to be the real deal.
Okay, so we've got a tool that's easy to use and has a really smart engine. But what does that actually mean for a researcher in their day-to-day work? Let's take a look at the journey from just simple data to an incredible discovery. For the researcher, the process starts with something like this. It's just a basic data table. You've got genes in the rows, different samples in the columns. No complex coding, nothing intimidating. They just upload the file. And from that one simple table, DGR works its magic. It generates a whole suite of these beautiful, insightful visualizations. You get things like heat maps that let you see patterns of gene activity at a glance, and these complex network maps that show you how different genes might be talking to each other. It literally turns a boring spreadsheet of numbers into a story a scientist can actually read.
But it doesn't just stop at pretty pictures, and this part is so crucial. DGR takes it another step with something called downstream analysis. It doesn't just hand you a list of important genes and say, good luck. It tells you what those genes actually do by linking them to known biological processes and pathways. This gives researchers instant context and a huge head start on figuring out what to do next.
So the tool is easy. It gives you great results. But the real question, the million-dollar question, is how does it actually stack up against the old, established, complicated methods? Well, this brings us to the DGR advantage. So get this, when they tested DGEAR on a common type of genetic data, microarray data, its results had a 94% overlap with one of the standard trusted tools. 94%. That's incredible agreement, and it proves that this easy-to-use tool is just as accurate as the old gold standard. But here's the thing, and this is where it gets really good. Being just as good isn't the whole story. The true power of DGEAR isn't just in agreeing with the old tools, it's in finding the critical stuff that those other tools completely miss. And here it is, the knockout punch. When they analyzed a more modern type of data called RNA-seq data, DGR found that a whopping 24% of the important genes it identified were completely unique. They were totally 100% missed by the standard tool. Think about that. A quarter of the potential discoveries were essentially invisible. Until now. And these aren't just some random, unimportant genes. No way. The unique genes that DGR found are involved in fundamental biological processes. We're talking about how our cells get nutrients, how they stick together and specialize, critical communication pathways, and even the drivers of disease like oxidative stress and inflammation. Finding these is a massive leap forward. And this really brings us to the core of what DGEAR is all about. The researchers who created it said it best themselves. Their goal was bridging the gap between computational complexity and user accessibility. It's about taking all that power out of a confusing black box and putting it directly into the hands of scientists. So when you step back and think about it, DGR isn't just analyzing data. It's empowering discovery. It's making the whole process faster, more accurate, and accessible to so many more experts. And that leaves us with one final, really exciting question to ponder. By making science simpler, just how many more discoveries are now within our reach?